So Unit 8 is about the bacteria, virus, or pathogen causing disease in us. So like I just told you, we're going to let them win for now. All right? Now, normally, we don't just let them do that. We, of course, normally attack them. And hopefully, for your own good, you win the war. So for now, we're going to let them attack us, let them infect us, and talk about the steps the pathogens have to do to succeed in causing disease in us. And it's really very straightforward here. So it first starts out with a discussion on something called your normal flora. It is also called your resident microbiota. Resident microbiota, normal flora, some places call it normal biota. It actually goes by many names. So normal flora, resident microbiota, what is that? It's simply all the bacteria that live in you and on you. That's what's considered your normal flora. Now, a lot of these bacteria, not just bacteria, other microorganisms, fungi as well. So a lot of these organisms are actually beneficial to you and I. Your normal flora is actually beneficial to you and I in many ways. And we'll talk about those ways. So what's an example of a normal flora? The streptococcus in your throat. That's an example of a normal flora. So some people, some students or people, they get infected, they get strep throat, and then they say this: I don't know where I got it from. It's been there. It's there all the time. It's part of your normal flora. The only time it becomes a problem to you and I is when they multiply out of control to the point where they do cause a problem. But if I sample your throat right now, most likely I will get streptococcus out of it because it's part of your normal flora. So in different areas of your body, you have different microorganisms that normally thrive there. And they live there because you provide them something, but they also provide you something for, at least in some cases, and we call that, if you remember, mutualism. So the normal flora in your gut, they provide you, for example, vitamins. Do you provide them food? They pay you back by providing you some vitamins. So, and again, we talked about that, we called it mutualism. Now here's another thing your normal flora will do. They will protect you. They will protect you against pathogens. Now watch this. One way they protect you is they keep your security guard wide awake all the time. And who would that be? Your immune system. Imagine if you hire a security guard to secure a building, and you walk in and you find them sleeping. That's not a good security guard, because you probably will lose everything, because people will come in, rob your store or whatever, while the security guard is sleeping. You want a security guard that is wide awake, red alert, always walking around checking for problems. That's your immune system. Your immune system is referred to as being competent when it can do that. So one way to make it or keep it that way is your normal flora. Your normal flora will keep your immune system on red alert. So it'll be working for you. So it's part of keeping your immune system active, healthy, and working for you. The other way it will protect you is through what is called microbial antagonism. What is that? Beautiful, there it is. When your normal flora that normally lives in your gut, for example, finds an invader and says, what are you doing here? This is my home. And they will antagonize them, meaning somehow they will kill them. So you may have eaten something that has this pathogen in it, and it eventually ends up into your gut. The normal flora that normally live there will attack that invader, and you wouldn't even know that you went through this. And that's what your normal flora is doing for you right now as you sit in the chair listening to it. Through microbial antagonism, it gives you the ability or the opportunity to say, I feel good. I'm not sick. I'm not worried about an infection, anything, because my normal flora in my gut, through antagonism, is protecting me. So this normal flora is very important for our health. Did you know, well, I guess now you will know, that your normal flora in your gut has an effect on things that happen in your brain? Research has shown that your normal flora growing in your gut actually has an effect on things like depression, anxiety, attention deficit disorder, among other things. It's amazing. Recent research has shown that your normal flora can also have an effect on obesity as well. And the more they study it, the more things will come out. Now, how does the bacteria living in my gut affect my brain? I mean, how does that happen? Well, when you learn in physiology, you learn that there's a vagus nerve that comes down. And the vagus nerve eventually reaches the gut area. The secretions from the bacteria affect the activity of the vagus nerve. Now, in some cases, it affects the activity in a good way. In other cases, it affects the activity in a negative way. Obviously, you want to balance it so that you get the good way. And nowadays, they tell you the pathway to health is balancing your normal flora for a lot of things. So you may have to talk to a nutritionist, your doctor, or whatever. What is it that you need to do to maintain a normal, balanced, healthy, normal flora? And we'll see if we can talk about it in another unit. So read about it. After the class is over and you're done with it, read about the benefits of balancing your normal flora to your overall health. Yeah.
Isn't that why there's a correlation between people who have like anxiety and them having also stomach issues?
Possibly, yeah. The vagus nerve will be influenced by the secretions from your bacteria. Now you're hoping that it's influenced in a positive way. And that can alleviate a lot of the issues that people have. And maybe the person can do away with medication altogether by simply balancing their normal flora. So read about that, look into that. All right, on that exam 1, there was a question asking you, if I remember, it was question number 2, how many BSL levels are there? And the answer is 4. The unfortunate thing is not too many of you knew that. And you'd be amazed of some of the answers. I think I had one student say 65. 65. I have no idea where they got that from. All right. There's only 4. In this lab, we cannot go beyond BSL-2. We are not equipped to use anything that falls under the category of BSL-3 and definitely BSL-4. No way. This room would have to be completely redone. And you can't just walk in here. You would have to suit up before you walk in here. And suit up and you'd look like an astronaut. That kind of thing. With oxygen hoses coming from the ceiling and all that. So there's no way we're going to use BSL 3 or 4 pathogens in a teaching lab. There's no way. They're too harmful for students to make a mistake with, and we're not equipped with. This always reminds me of something that I went through. A long time ago, I went ahead and looked for another job. And that was a long time ago. And I found one at the Centers of Disease Control. Looked really good, really good. That salary was good. I applied for it, and I got it. And then I said, hang on, let me ask him something. And it was a ridiculous question. What's the highest BSL that you use at the CDC? If there was a BSL 5, they would be using it. You see what I mean? They looked at me like, all of them? Okay. The reason why I asked is, I thought about, hang on, in all the years of research prior to that, how many times did I drop something? I said, many times. I called them back and I said, thank you, but no thank you. I drop one of these on me, I'm not going home. And if I do go home, it runs a risk of me and potentially my whole family being wiped out. Maybe my whole block. You see what I mean? And I realize that I make mistakes. Sometimes I drop something, but that's not something I want to drop. I said, you know what? It's not worth it. So I decided to call them back and say, no, thank you. And I found something else to do. Now, I'm not saying that you should shy away from something like that. If you get an offer like that, think about it, entertain it, but know that you're going in with that slight level of danger, obviously, that you need to be aware of and practice means of protection, of course, with your lab coat, goggles, and so forth. And this goes beyond that. This thing here, after you're done working, besides the fact that you're in the suit the whole day, and that's another thing I did not want to do. You have to walk out into another room and stand under a shower, and it's not necessarily water coming out of there, and wash the suit and all that. I don't need to do all that. So at least to me, it was not worth it there. All right, some words that we need to become familiar with. The word pathogen. I think we all know what that is, a microorganism that causes disease. There you go. But these two words, true pathogen versus opportunistic pathogen, tends to be a little tricky for some students, at least initially. Listen to it carefully. A true pathogen is a pathogen that will cause disease in you, even if you're immune competent. That means I ask you, how do you feel today? I'm fine, I'm good. How's your immune system? It's perfect. And then you walk out and you get sick. You got sick from a true pathogen. A pathogen that causes disease in a normally healthy individual. And this is typically what we think about when we think about pathogens. We say, I don't want to come down with an illness. That means you're already fine. So you try to stay away from those true pathogens that make you sick, even though the split second before you came in contact with them, you were healthy, you were fine. You will actually understand true pathogen a little bit more when I explain to you the opportunistic one. An opportunistic pathogen is a pathogen that becomes a problem, of course, when your immune system weakens. So it takes the opportunity that your immune system is weak. So your normal flora can become opportunistic pathogens. So for example, when you take an antibiotic there's a very good chance by simply taking the antibiotic, you become immune compromised while you're on the antibiotic. And another pathogen takes advantage of that. That's an opportunistic pathogen. And we talked about it before when I talked to you guys about gastric ulcers. You have to take the antibiotic to get rid of the H. pylori in your stomach. But that's a strong antibiotic. It will weaken your immune system. And the fungus in your mouth, you have that in your mouth, the candida in your mouth, it's normal flora. They normally don't do anything to you. They're just there. But it will take the advantage of you being weak, and it will become an infection. And you'll see a white film on your upper part of your mouth and on your, especially on your tongue. So a doctor giving you the antibiotic to take care of your H. pylori will also give you an antifungal, not for the bacteria, it's for the fungus, that he or she knows you will get 2 or 3 days after you're on the antibiotic. That's an opportunistic pathogen. You know what an opportunistic pathogen reminds me of? Your average teenager, 15-year-old, when mom and dad are home, yeah? Your average teenager is behaving well, great, awesome. He's in his room studying, maybe praying, sleeping, but not causing problems. Great. He just waits until mom and dad leaves home. When mom and dad leave home, that's your immune system weakening, you see? And what happens? Your son calls everybody. Hey, mom and dad left town. Party at my house. Everybody comes over. The next door neighbor and the faraway neighbor, you see? Mom and dad finally come home to a burned down house. That's an opportunistic pathogen. Waits until you're immune compromised and causes a problem to you. Very good example or analogy for that. Now you gotta understand something. Where was your son? He was already in your house. He just waited for you to leave. Endogenous infection. Where's your normal flora? It's already inside you. All it's waiting for is your immune system to weaken. Do you see that? So these two go hand in hand. Who is the culprit of an opportunistic pathogen? Your normal flora. They're already inside you. So that's called an endogenous infection. Now this part, a lot of students understand this, but now they forget this part. And I will probably ask you this in the unit questions, potentially on the exam. Listen to it now. There's another way a pathogen can be opportunistic, besides you being immune compromised. And what's that way? Listen to it. When the pathogen finds itself in a location where it doesn't belong. So something like E. coli. There's E. coli in your gut. They're actually helping you do a lot of things. Digest your food, vitamin K, microbial antagonism and everything. And you're not going to worry about them being there because actually you need them there. But E. coli in your blood, you got a problem. You got a huge problem. It will potentially lead you to sepsis and septic shock. You need a professional physician to save your life. So what happened? The E. coli that was in your gut, found itself in a place where it's not supposed to be. And you definitely would need a physician that can pick up on that and treat you the right way, because a lot of times once you approach septic shock, it's very difficult to reverse. Another example of that is the bacteria in your appendix. The bacteria in your appendix, as long as they're in your appendix, you're fine. Actually, in some cases, you need them there. But if your appendix inflames, swells, usually you feel pain, usually. And it's excruciating pain. I don't think anybody can ignore that pain, no way. But let's say you do, for some weird reason, and it bursts on you. Go ahead, call 911 and see who's going to help you. They most likely, by the time they get there, by the time they figure out it's an appendicitis, and then by the time they get you to the ambulance and to the emergency room, whatever, you should have addressed it early on when you felt the pain initially, not after it burst open. But now what happened? The bacteria that's in your appendix is now in your abdomen. It's in a location where it doesn't belong. So 2 ways of being opportunistic. Immune compromised, right? And find yourself, the bacteria finds itself in a location where it doesn't belong. Okay, the process, the disease process. Remember we said that we're going to allow the bacteria to win in Unit A. The first thing the bacteria is going to do, bacteria or virus or whatever, is gain entry into you. Now the normal flora, they're already in you. So we're typically here, when we talk about portal of entry, we're more talking about the exogenous infections, the infections that you obtain from outside. So the pathogen on the countertop or in the air or whatever in your water has to gain entry, and that's called portal of entry. And then it goes through, and we'll talk about these things, attached to the surface of your cells. It has to survive your attack on them. They will now start causing illness, disease. That's when you start feeling their pain. And then they will leave and say goodbye to you. Portal of exit. Why? Because they want to hurt somebody else. They're done with you, they want to affect somebody else. So those are the 5 steps. Portal of entry, anything, any opening you have is a portal of entry, obviously. Your nose, your eyes, your ears, your mouth, vaginal, everything. Everything is a portal of entry. If it can gain entry, it will use it. The easiest one is your lungs, breathing. Some of the most successful pathogens, the ones that usually cause things like pandemics and stuff, are things that we breathe in. I mean, think of COVID-19. Why did COVID-19 succeed so fast, transmitted so fast? Because all you have to do is breathe it in, and then later you sneeze it out. So one sneeze in a mall will hurt everybody. Why? Because it goes into the vents and it starts circulating. That's why they told us to wear masks. So pathogens that linger in the air, they tend to be very successful in causing problems, because they know you're going to have to eventually breathe. And if the pathogen is right here and I breathe, I just took it in. Now, good luck to me. So respiratory, easiest way to get in. Now bleeding, if a pathogen is transmitted through blood, for example, well, that kind of limits the people that will come in contact with that. An infected person would have to come in contact with possibly a police officer dealing with them, an emergency medical technician, a nurse, a doctor, So now you're limiting maybe their husband or wife or whatever. So you have to have direct contact with the blood to have a problem. The respiratory one, I can sneeze and then leave. 5 hours later, you come in, you breathe that in. There you go. I don't have to be there when you're doing that. But bleeding, that tends to be more of a direct contact, so less effective that way. But it's possible. So any opening is a portal of entry. So that's what the pathogen is going to do. Gain entry. Now what? You know, once they enter, they know what you're about to do. You're about to wash them out. And of course, what are we saying about Unit 8? We need them to succeed. So, what the pathogen is going to do, watch this. They know you're about to wash them out. So they're going to do this. Whatever they have, they're going to do this. Watch. Attach to you. Let's say this is your cell. They will attach to your cell. And then they will look back at you and they say, wash me now. Try to wash me out now. You can't. Why? Because I'm stuck to you. And that is the next step. Attach to the host. Now, what are they going to use? Well, it depends on who you're dealing with. Some bacteria will use their fimbriae. That should come in. You should remember that from the previous units. Fimbriae is used for attachment. If they have fimbriae, that's what they're going to use. The capsule can be used for attachment. If they have a capsule, they'll use it. The biofilm, the slime layer, can be used for attachment. That's basically like the plaque on your teeth. The plaque on your teeth is attached to your teeth so strongly that obviously your normal brushing doesn't work. You need to go get professional cleaning to dislodge that with ultrasound and all that stuff. If you're dealing with the virus, they're going to use their spike proteins. And you should be familiar with that. Spike proteins adsorption. So, let's review. Portal of entry. By the way, I forgot to mention something with the portal of entry. Your skin is a portal of entry. But then you're looking at your skin and like, how is that a portal of entry when you cut it? You see what I mean? Intact skin, no. Actually, intact skin is protecting you from a lot of things. Because what was the first thing that we learned in this lab? Bacteria are everywhere. What was that word called? Ubiquitous. And your skin is protecting you second by second for the rest of your life. Until you cut it. Once you cut it, there we go. There's a portal of entry. And we'll talk more about skin and other things in the next unit. So we do have what's called physical barriers. Your skin is one of them. Our secretions is another one. Our tears, our saliva, our stomach acid, these things are all collectively referred to as host defense mechanisms. And we'll talk more about this in Unit 9, all the different host defense mechanisms. You walk it out? Yeah, walk this way. All right, so the host defense mechanisms include tears, saliva, stomach acid, your skin, and your nose hair, your mucus. We'll talk about all that in Unit 9. So again, in Unit 8, we want the bacteria or the pathogen to succeed, right? So they have to overcome the defense mechanisms. They have to defeat your defense mechanisms if they want to survive, of course. One of the most important host defense mechanisms is phagocytosis. There are professional white blood cells circulating in your blood right now. That's their job, and that's why they're professionals at it. For as long as you've been alive, that's what they've been doing. They've been going through looking for bacteria. And once they find them, they phagocytose them. They eat them, basically, phagocytose them, and they kill them with the lysosome. And we'll talk more about that too in the next unit. So phagocytosis is very important. It's a very important host defense mechanism. Well, again, we want the bacteria to survive. So they have to produce things that protects them against phagocytosis. And that's these things, anti-phagocytic factors. So the pathogen will release or produce anti-phagocytic factors. So let's say, for example, she is coming to phagocytose me. I'm the bacteria. And she's a professional at it. Normally she would do it, and I would not survive. Unless I secrete what? Anti-phagocytic factors, and I look at it and I say, nice try, but not today. One of the best examples of an anti-phagocytic factor is the bacterial capsule. The actual capsule itself is an anti-phagocytic factor. If I had a capsule, she would have a very difficult time phagocytosing me. You see that? That means I would survive and cause an infection in you. So let's review. Pour to love, pantry, and then attach, and then survive the host defense mechanism. So I'm alive. Am I right? I'm the pathogen. I'm now alive. Now I start secreting things. One of those things is exoenzymes. For example, if I was Staphylococcus aureus, I would secrete coagulase. You should remember that from the lab. And then take your blood proteins, remember that? And build a wall around me to protect me from your immune system. So one way to defeat the host defense mechanisms, is to build this barrier around me. In case I don't have a capsule, I'll build a wall with your blood proteins, but I would have to secrete coagulase. So because I'm secreting this coagulase to the outside and it works outside, that's called an exoenzyme. An enzyme secreted by the bacteria and it works outside is called an exoenzyme.
Is it common for like gram-positive bacteria to have this mechanism since they don't have a capsule?
I wouldn't worry about the whether they're gram-positive or gram-negative. I would worry about if they're able to secrete the coagulase or not.
Okay.
Yeah, see, I wouldn't worry about that detail. But if they're able to secrete the coagulase, they will take your blood proteins and build that barrier, protecting themselves from your immune system. They survive the host defense mechanisms. Then they start going, after that, they'll really go to work against you. And because they're secreting the enzyme to the outside, that enzyme falls under the category of an exoenzyme. Another exoenzyme is the hemolysin. But that now is for a different purpose. The hemolysin secreted by bacteria, they're after the iron in your blood cells. And why would they be after the iron? Because they need it to stay healthy. So that's a different approach now. They will go and get your iron, so they can be healthy. And if they're healthy, they hurt you more. Do you agree? So slightly different way of protecting themselves. They just want to stay healthy. Yeah.
What's an example of a disease that uses hemolysin?
Oh, there's strep throat. Streptococcus pyogenes that causes strep throat is beta hemolytic. Okay, what's another example of an exoenzyme that you used in the lab? Lipase. And what did lipase digest? Lipids. Very good. What's another example of an enzyme that we used in the lab? Starchase. What did starchase digest?
Wait, starchase or amylase?
That's what I'm joking. I'm
Oh.
joking.
That's
I'm
it.
waiting for someone to say starch. What did starchase digest? Nothing, right? Because there's no such thing as starchase. What's it actually called? Amylase. Very good. That was a joke. Anyways, how about DNase? DNA. What about the one that digested milk protein? What was that called? KCase. Very good. All of these are exoenzymes. They're secreted by the bacteria and they work outside. Then we get to this slide. This is the last slide for today. Then we get to this slide, we talk about something called exotoxins and endotoxins. Now I teach at another place, another university there, and one of the questions asks them to describe or compare and contrast the difference between an exotoxin and an endotoxin. Now, luckily for you guys, it's all multiple choice, right? But for that question, they have to actually write a paragraph on it. And some students, beautiful, beautiful answers. I mean, wow, I couldn't have done it better myself. But for most students, they don't study this, unfortunately. So they rely on test-taking strategies. Exotoxin? That's a toxin that's on the outside. Okay? Endotoxin? That's a toxin on the inside. Compare and contrast, and that's the answer? I can tell from the one sentence answer, that student has no clue what those two things are. Because that's not how you can, no way you can compare and contrast exotoxin and endotoxin using one sentence. Good luck. No way. And then sure enough, I read it and I'm like, yep, I knew it. They don't know what they're talking about. Exo, endo, that has nothing to do with it. To some extent it does, but that's not, it tells you right there, you don't know the difference between the two. Now listen to it. An exotoxin is a toxin, yes, it is secreted to the outside. That's fine. But that's not the full story. The full story is this whole table. And I know for a fact you can't fit the answer of this entire table in one sentence. No way. So I can tell, no way. One sentence, no way they have the answer for that. So listen to it now. An exotoxin is a toxin secreted by a living bacteria. The bacteria has to be alive, obviously, to secrete this toxin to the outside. But now you have to understand what the toxin is. It's a protein. The exotoxin is a protein that's secreted by the bacteria. This protein can eventually find its way into your blood. Now you have a big problem, a lot bigger than the infection. Have you ever heard this statement? If not, you'll hear it now. A healthy mouth is a healthy heart. Have you ever heard that before? Yeah. Well, now you heard it. Talk to your dentist about that, and they'll ask you, that's pretty good, you know about that. Yeah. What happens? Watch this. The bacteria in our mouth, meaning in our teeth, and if we're not cleaning it on a regular basis, ignoring it, whatever, besides the decay and all that, you have a potential bigger problem. What is that? The bacteria in your teeth can secrete exotoxins. into your blood. And then now they're going through your body, and some of the exotoxins target your heart cells. Now, they don't touch anything related to, for example, your liver, your kidney, whatever. No, they target your heart. Your heart cells have receptors that happen to bind to these toxins. And they're toxins. They hurt these heart cells, of course. So a healthy mouth is a healthy heart because of these effect of these exotoxins. Now, some other bacteria may release other exotoxins. And the other ones can hurt your lungs, for example, or hurt your liver, for example. So now we understand how important these things are. Exotoxins are proteins secreted by the bacteria, and they hurt your cells. They can actually kill your cells. That's why they're called toxins. They are transported through your body, through your blood. But they target specific tissue, depending on which toxin it is and which receptor this tissue happens to be expressing. Now watch this now. Because they're proteins, they're heat labile. That means you can denature them with heat. Because they're proteins, you can denature them with heat. That's the good side. That's the good news. Watch this now. There's another thing that you need to know. Because they're proteins, they're also antigenic. What does that mean? That means they can stimulate your immune system to produce antibodies against them. If you survive the first attack. You see that? Watch this. You get a bacterial infection. It releases an exotoxin. Your body recognizes that as foreign, and it will produce antibodies against it. Now, let's say you survive the first infection. The next time you see that same pathogen secreting the same exotoxin, you'll be protected because you'll have antibodies against it. And that's why I said, if you survive the first initial attack. But there are some exotoxins that if you get them, you don't survive it. Now what do we do now? How do we protect you? Watch this now. They grow a large batch of that bacteria and then isolate the exotoxin in the lab. So we're going to isolate the exotoxin in the lab because this exotoxin is very harmful to you, to the point where you won't even have a chance to make antibodies against it because you'll be dead from it. So to protect you, what they do is they take the exotoxin from bacteria grown in the lab, they isolate it, they purify it, they denature it. Why can they denature it? Because they are proteins. And once they denature it, they take it, and inject it in you. Because it's not effective anymore, it's denatured. But what will it do? It'll stimulate your immune system. So that the next time, God forbid, you do get exposure to the real toxin, you're already protected. So they're using it as a vaccine. They take the denatured exotoxin and they use it as a vaccine to stimulate your immune system to make the antibody, because if you do get the real thing without the antibody, you don't survive that. Obviously, there's a name for everything. The denatured exotoxin used as a vaccine is called a toxoid. Toxoid. That's when I take the exotoxin denature it on purpose, inject it in you on purpose for vaccination purposes. The outcome is you produce an antibody, which would be called an antitoxin, and now you're protected in case you come in contact with the real thing. Yeah.
So I have a question.
Mm-hmm.
My question is, can that relate to autism in kids?
Can that relate to?
Autism in kids?
I don't know about that.
Because isn't there like something in between that?
There's research that says yes, and there's also research that says no,
Okay.
which means we don't know. Yeah, and actually, it might not be the actual vaccine that might be causing that. It might be the additives. So they have to add things to it to get your immune system to stimulate and all that stuff. It might be associated more with the additives than the actual main components of the vaccine. But the real story on that is out. We don't really know. There are some studies that say yes, some studies that say no. Yeah. But there's a lot of benefits of obviously vaccination. All right, so that's the exotoxin, and you can tell you can't write that in one sentence, of course. Now, what is the endotoxin? This one now has nothing to do with the endo part. An endotoxin is actually also something that's going to be secreted into your blood. So it has nothing to do with the endo part. So what is it? An endotoxin is basically pieces of the outer membrane of gram-negative bacteria. So now watch this. As long as the gram-negative bacteria is intact, that's not going to release an endotoxin. Look up here, please. As long as the bacteria is intact, that means it has an intact outer membrane. That's not going to release an endotoxin. This guy has to die. So unlike this one, has to be alive. The gram-negative bacteria has to die and pop like a balloon. And what will happen? The outer membrane will fragment. It will release millions of pieces of lipopolysaccharide. The endotoxin is the lipopolysaccharide. LPS of gram-negative bacteria, but not the intact outer membrane. It has to do this. The bacteria has to die, and it has to burst open like a balloon, and the outer membrane has to fragment into little pieces of LPS. This is the endotoxin right here. The little piece of LPS. So obviously the gram-negative bacteria has to die. Now, can I use the LPS to protect you like I did with the exotoxin? No, because that's not a protein. The LPS is lipopolysaccharide. It's a lipid with a carbohydrate that is not antigenic. Even if it does stimulate your immune system, it'll be very little and not even worth it. Why? Because actually, if I inject LPS in you, you now have another problem. What is that? Massive inflammation. I won't protect you. No, I probably will kill you. LPS will cause a massive inflammatory response in you and I. And that's how it hurts you. And it's not the outer membrane, it's pieces of the outer membrane. That means the gram-negative bacteria has to be dead to release it. So that's why I said that endo part is deceiving. It has nothing to do with inside the cell versus outside of the cell. No. So the endotoxin is the lipopolysaccharide, and go ahead, try to denature it with heating. Not effective. It is heat stable. It's almost completely opposite of that of exotoxin. Whatever exotoxin is, the endotoxin is almost completely opposite of that. Can you describe this in one sentence?
No.
Of course not. That would actually be an awesome question to add to my Valencia students too. But not this semester. Sound good? You got enough problems, right? One problem is the exam on
Thursday.
Thursday. Go home and do it.